Okay. Good evening, everyone. Students, I decided to make a couple quick videos to help explain some of the things that you've been learning in class. Unfortunately, some of it on your own. Anyways, thought I'd use some of this new technology and sort of, let you in on a little bit of the wolf life. So I think Carson just went to bed a while ago and see some nice decorations and whatnot in the kitchen here. So I'm going to go through a couple different things, review some stuff, hit a couple new topics for you, and hopefully this will help you understand things. Hopefully getting back to school soon. Anyways, see if you can learn even with me not there, which I'm sure you can. So we'll do some biology here. I know you'd probably like to sit and do whatever, but the learning must continue. All right, so let me pull up a couple things here. Pardon me a second here while I bounce around some. Okay, so I want to go over this with you again real quick because this is something that you guys have been looking at and learning about. Let me try to pull up a pen here so I can write test here. All right. So anyways, this is a picture here of the Lac Operon system that you've been learning about. And I guess let me back up a second here. Pardon me. Yeah, so what this is talking about is that regulation of a gene, in other words, turning it on or turning it off, can occur at some different times. And the two main times that we want to talk about are what's called pre-transcriptional or post-transcriptional. Pretty self-explanatory. Pre-transcriptional would be basically to stop the gene or to start the gene at the transcription stage. Is it going to be transcribed or is it not going to be transcribed? And then post-transcriptional is pretty obvious also. Be able to turn off the gene or to keep it from being translated even after it's been transcribed. So you'd have a piece of messenger RNA that would be able to go to the ribosome and be transcribed, but for whatever reason, it's not. For whatever reason, maybe it doesn't get translated. And this would occur in eukaryotes only. You wouldn't see post-transcriptional regulation happening in prokaryotes. Bacteria. This would only happen in eukaryotes. So, all right, let's bounce ahead here a little bit. So back to this Lac Operon system that you've been learning about. So again, this is a pre-transcriptional regulation mechanism. And again, what you have here is you have the piece of DNA, which is this long ribbon here in this picture, and you have the structural genes down here, which are 3 sections of DNA. They're marked Z, Y, and A here. And these basically, when they're transcribed and translated, produce enzymes that digest lactose, which we've talked about quite a bit. This big molecule right here, sitting here, is RNA polymerase, and this thing would like to go over here and transcribe these 3 genes, transcribe this DNA into messenger RNA that's over here. But in this situation, it can't do that because this giant repressor protein is sitting here. It's blocking the RNA polymerase from getting to the genes to transcribe them. So in this situation right here, this is where we say it's turned off. It's not going to be transcribed. A couple other things I want to make sure you know. So the spot on the DNA where the RNA polymerase binds is called the promoter, and the spot where the repressor binds is called the operator. So some things you definitely want to understand and know here is, understanding that this is all DNA, and there's genes down here. You have the operator site where the repressor attaches, the promoter site where the polymerase is going to attach. I guess understanding that the repressor is blocking the RNA polymerase from getting down here to the genes that will be transcribed and translate into enzymes that will digest lactose. So when would you want this to be turned on? Get back here. You'd want this thing to be turned on. If you're a bacteria, you'd want this to be turned on when there's what around, and the what is lactose, and lactose is around. So in this picture down here, this little heart-shaped thing is basically lactose. And what lactose does is connects to this repressor, causes the repressor to fall off the DNA. And when that happens, this repressor, the RNA polymerase can then move down here and transcribe these 3 genes here. And these will become messenger RNA that will go off. Let's see if I can write on here. It's not too bad. That RNA would go off to the ribosome then and be translated. So this is basic control in a prokaryote. How prokaryotes regulate their genes. Okay. Finally, the last thing I want to make sure you understood and knew is something called post-transcriptional gene regulation. It's obvious it's something that's going to happen after the RNA is produced, after the gene's been transcribed. A lot of crazy stuff that can happen here. It's pretty interesting. Let me jump ahead here. Actually jump around. Okay, so we talked before about this thing called the Human Genome Project, which was the effort by humans to sequence the entire human DNA, and this started back in the 90s, early 90s. Interesting thing, so when they started the project, they knew there was about 100,000 genes, 100,000 proteins that occur in the human body. So understanding how things work, transcription, translation, everybody expected to find 100,000 genes because there were 100,000 proteins. About halfway through the project, kind of startling news, people working on this announced that they thought there was only going to end up being about 50,000 genes that made up the human genome. Finally, when it was all finished, shockingly, again, it turned out that there were only 23,688 gene coding regions in the human DNA. So hopefully this causes you to ask a question, which would be, how can we produce 100,000 proteins from only 23,688 genes? Doesn't make any sense. So obviously people started to look at this question and try to figure out what was going on. So jump back here. One of the things that allows this to happen, and I'll come back and explain this a little more, but since I'm right here, something called RNA editing, or post-transcriptional changing of the RNA. So it'd be after it's transcribed, there's some things that can happen to it that can allow it to actually be used to make multiple proteins. This is just one example. There's a single gene coding region in the DNA that has been associated with producing 11 different proteins. This occurs in the brain. And so again, the obvious question is how? How does that happen? And so what I want to show you is something called exon shuffling. And I think I'm about out of time. So I'm going to end this one, and we'll talk about exon shuffling on another video. See you.